I'm excited to introduce Ben Kleinstiver, an assistant professor at the Massachusetts General Hospital and Harvard Medical School.
Alrighty, thank you so much for the invitation. Hopefully the slides and audio are coming through okay. As mentioned, my name is Ben Kleinstiver. I run a research lab at Mass General Hospital. And one of the major focuses in my lab is to think about ways that we can develop new and improved genome editing technologies. And I'll tell you all about that today. So quickly, just a disclosure of some outside activities. I'm an inventor on patents, including work that I'll tell you about today. And then I advise and consult for a few different companies. So as you know, and I think you just heard, CRISPR technologies really are enabling really wonderful applications, both in biomedical research and for human therapeutics. And I think this slide nicely shows how customizable the types of edits are that we can make to the genome, all built on this single chassis of Cas9. We can use CRISPR-Cas enzymes as nucleases to knock out genes. We can use sort of next generation genome editors like base editors or prime editors to correct and write genes, and we can use epigenome editors to regulate genes. I'm getting a question, are the slides moving? Hopefully. Hopefully you're seeing a slide titled Properties of Genome Editing Technologies. So, you know, with all these genome editing technologies that we have at our disposal, I think we like to think about the fundamental properties of these technologies and how effectively they work sort of out of the box, out of naturally occurring systems, and then think about ways that we can improve them to make them more effective genome editors. And one of the most important properties that I think matters to the genome editing community is the on-target activity of an enzyme. Essentially, the question is, how well can this protein edit at a given site? Another really important property is the targeting range of an enzyme. The question there becomes, where in the genome can it edit? Are you able to position the edit precisely where you need to make the exact change that you intend? Another really important question is the specificity or safety of the genome editing technology. The question there being, where else in the genome is the technology binding and editing? And are you making other off-target edits in the genome that could be problematic for your approach? And then finally, other properties like the edit outcome. Genome editing technologies rely on some of the underlying DNA repair pathways in cells, so they can make lots of different types of edits. And usually a researcher or a clinician wants a single exact edit in their population of cells. So I think, you know, a big effort in the field is devoted to how we can develop CRISPR enzymes that are tailored to making very specific types of changes. And then finally, there are lots of other properties that, you know, we have to consider both for research and therapeutics. One being delivery, others being, you know, the immune reactivity of these enzymes. But as a whole, I show this slide just to highlight the many different ways that we can think about improving these technologies. So with that context, I also like to think about what would an ideal genome editing enzyme look like. Obviously, you'd like it to be highly active so that it edits a high proportion of cells. You'd want it to be able to access any target site in the genome so that it can edit wherever you'd like. And of course, you'd want it to be safe so that it edits only at the site that you target it to and doesn't make edits anywhere else in the genome. And I think if we think back to where these CRISPR enzymes came from, they originated as a host event system in bacteria. Totally different requirements in that system versus what we want as a research reagent. And in fact, all these properties that I highlight on the top of the slide, these are actually contrary to the fitness of a bacterial immune system. So I think this has major implications for the clinical use of CRISPR enzymes and how we can take these enzymes out of bacteria and repurpose them to be used effectively as research reagents or in humans. So that's where we use protein engineering actually to try and solve this problem, where we can take these naturally occurring enzymes and tweak them to make them more amenable as human therapeutics. So as an outline of what I'll tell you in the next 30 minutes or so, some of the major goals in my lab are to develop more broadly useful tools. And I'll tell you one story about how we've been able to take one CRISPR-Cas enzyme and engineer it to make it able to read essentially the entire genome. Another major focus is how we can design safer technologies where CRISPR-Cas enzymes naturally do have off-target effects. And what we've been able to do is engineer versions of these enzymes that now have better specificity, only targeting their intended site. And then finally, I'll talk about some of our efforts to explore how these new expanded enzymes enable us to perform applications that we simply could not before without them. Okay, so one of the fundamental questions that we think about in the lab is where in the genome we can edit. And this is essentially the question of targeting range and how this can be problematic for various genome editing applications. For CRISPR-Cas enzymes, this is dictated by this property called the PAM. And this stands for protospacer adjacent motif, which is a little bit of jargon, but essentially this is a sequence in DNA that the Cas9 protein has to recognize to be able to bind and edit in the genome. So where there is a PAM, the Cas9 enzyme can essentially land on that DNA sequence, unwind the DNA strands, and make an edit. But in the absence of a PAM sequence, the Cas9 enzyme doesn't stick around long enough on that portion of DNA to make an edit. So the PAMs dictate where we can make edits in the genome. Now bacteria, they actually use the PAM as a proofreading mechanism so that they don't edit their own genome. They use the PAM as a way to identify invading DNA so that they can cleave invading DNA, but they don't have PAMs in their own genome, so that prevents them from self-targeting and essentially destroying their own genome. But as we repurpose these tools for genome editing in human cells, again, the availability of PAMs dictates where in the genome we can target. So we can only make edits where we're able to identify PAMs that are accessible by these Cas proteins. And what that means is that the regions between the PAMs actually are largely inaccessible for editing because we can't target them effectively with naturally occurring CRISPR enzymes. So one of the issues then is, as I showed you on the first slide, we have all these amazing things that we can do to make edits or functionalize the genome. In the absence of PAMs, we actually can't perform these applications very precisely. So hopefully this highlights the need of having enzymes with improved targeting range. So what we then did with a sort of problem in mind is we thought about CRISPR-Cas enzymes and how they actually recognize the PAM. So here's a crystal structure solved by Martin Yinnick's group, showing the Cas9 enzyme in gray and a target DNA sequence in black here. And essentially from this crystal structure, we can understand what amino acid side chains of the Cas protein are important for recognizing the PAM in the DNA sequence. So highlighting a few of those regions of the protein here, we can zoom in on some specific side chains of the Cas9 protein, knowing that these are really important for reading the PAM DNA bases. And by knowing this, actually, we can begin to formulate engineering strategies about how to change how the Cas proteins recognize the PAM and potentially removing that PAM requirement. And to do this, we really wanted to ask this question in a high-throughput way. So I'm showing you data from an older study during my faculty transition period where we were engineering a completely different CRISPR enzyme. Essentially, Russell, who was working with me at the time, purified 12 different proteins to try and understand what amino acid side chains are important for PAM recognition. And for anyone that's ever purified one protein, to purify 12 takes 12 times as long. This is not a very scalable process. And what we wanted to do is identify this as a bottleneck in this process and be able to eliminate that so we could engineer at a higher throughput scale. Essentially, I don't think anyone has ever wanted to purify hundreds of proteins to do this type of experiment. So what we wanted to do is come up with a system where we no longer needed to purify these proteins to understand their properties, but instead have a proxy system so we could understand the PAM requirements of different Cas enzymes in the absence of having to purify them. So we came up with a system, and this is a system that allows us to scalably understand the properties of these Cas proteins as a way to then begin to engineer their properties like their PAM requirements. So instead of purifying these proteins one by one, what we can do is we can essentially clone a plasmid encoding the enzyme, transfect it into human cells, which we can easily do in 96-well plates, So we can do this essentially 100 enzymes at a time. We deliver the enzyme into cells. We normalize the concentration of the protein based on a GFP marker that's co-translated with the Cas enzyme. We take that lysate of the Cas protein, and then we can use that for in vitro biochemical assays. And if all of this on the slide is jargon, this is essentially just a workaround that makes us not have to purify each protein individually. We can study 96 proteins at a time because we can transfect them into human cells, lyse these cells, normalize the amount of protein, and then do some biochemical assays. So this assay actually allows us to think about understanding Cas enzymes in a different scale, going from 1 protein at a time now to almost 100 at a time. So using this system now, this HT-PAMDA assay or this high-throughput PAM determination assay, we can then use this system to understand how we can change Cas9 to modify its PAM requirements. So I'm showing you a subset of some of the data that we can generate with this assay. Where here, a dark blue box means that the enzyme can effectively target this PAM sequence, and a light blue box means that it can't. So here on the top line, this is wild type SP-Cas9 that most people know and use. And you can see here the sites that it can edit are those with an NGG PAM, and this is just a subset of the possible nucleotides in the genome. We previously engineered a different version of Cas9 that we call VRQR, and this is just based on the changes in the amino acids. And you can see that its PAM requirement is actually quite different. It actually prefers NGA PAMs instead of NGG PAMs. And we can then use this HT-PAMDA assay to engineer lots of different versions of Cas9. Essentially now trying to relax the PAM requirement, we can go from either an NGG or an NGA variant down to this variant on the bottom here that now has a relaxed tolerance for PAMs that we called SPG, because it now required only a single guanine, meaning that it could recognize all these different PAM sequences. So we can essentially use this HT-PAMDA assay to profile the impact of these changes on the Cas9 scaffold, where again, the wild type Cas9 prefers this box here, which represents the NGG PAM space. But now this new enzyme you can see has a very different profile where it targets this entire row of NGN PAMs. Okay, so that's all in vitro work to show you what this means in human cells with these new enzymes. This is 78 sites, 78 different guide RNAs in human cells. You can see that wild type SP-Cas9 is largely only effective against the sites with NGG PAMs. But when we use this new enzyme called SPG that we built using the HT-PAMDA assay to characterize its properties, you can see now that this SPG enzyme has high activity against these sites that previously were inaccessible with wild type, especially on these NGC and NGT PAMs, where you can see essentially only activity with the blue bars with the SPG enzyme. So for the sake of time, I'll just skip ahead there. So we have this enzyme SPG now that allows us to access essentially 1/4 of the genome, as you can see here. 1/4 of PAMs are now accessible to this single enzyme. What we wanted to do then is use this SPG enzyme as a scaffold to further engineer it to see if we could access more of the genome. So we could go back and look at crystal structures of Cas9 and ask the simple question, could we make SPG, or could we make changes to SPG to relax its PAM requirement? So what we're able to do, again, using the HT-PAMDA assay, is make additional substitutions in some of these amino acids shown here to arrive at this enzyme that we call SpRY, that now can access the NA space of PAMs that were essentially inaccessible before to SPG. So in human cells, what that looks like now is we have this one enzyme that can access essentially all of these PAMs or target sites that wild type Cas9 was ineffective at editing before, meaning that we have this one enzyme now that can edit the vast majority of the genome. And this approach was largely enabled by having the scalable assays that allow us to do the engineering. So this work is published. I encourage you to check out the paper. Essentially, we were able to take wild type Cas9, remove a single base G requirement, arriving at this SPG variant now that requires just an NGN PAM, and then further engineer SPG to develop SpRY that essentially can edit almost all of the genome now with a single enzyme. And we think this is interesting because it not only does it unlock the genome for editing of essentially any base pair, but this also tells us that the Cas protein scaffolds are actually really amenable to this type of protein engineering to fundamentally change their properties. Okay, so I've told you how we've been able to take Cas9 and relax its PAM requirement to make it edit more of the genome. Now I'll just briefly talk about the safety of these enzymes. So the safety obviously is a really important consideration for how you use these enzymes, both for research and therapeutics. The specificity of the enzyme is largely dictated by the ability of the guide RNA to pair perfectly with the target DNA. But also the specificity is governed by the ability of the Cas protein to recognize a PAM specifically. And if you remove that requirement for a PAM, you're essentially de-engaging this initial licensing step of how the Cas protein recognizes its target site. So ideally, we want Cas enzymes that have high specificity and avoiding situations where they have off-target effects because they can have unpredictable or unknown consequences at the genome scale level. So we used an assay called GuideSeq that was originally developed by Shendar Sai while he was in Keith Jung's lab to look at off-targets in cells. And how GuideSeq works is we deliver the Cas enzyme into living cells. We also deliver the short double-stranded oligo tag. It gets captured into DNA double-strand breaks. And then through some molecular biology, you can identify where and how efficiently these GuideSeq tags get captured throughout the genome. And that allows us to map the location of on and off target editing sites. So on the top here, I'm showing you some data with wild-type Cas9 with a guide RNA, and you can see here the second line from the top. This is the on-target site with no mismatches highlighted. And then these other sites here are off-target sites. They're edited at varying efficiencies compared to the intended on-target site. And you can see the mismatches occur at different positions of the target site as well. So our hypothesis then is that by using GuideSeq to look at the specificity of these new enzymes is that as we relax the PAM requirement of SP-Cas9 to SPG, then to SpRY, we might observe new off-targets because again, the PAM requirement is the initial licensing step of how the Cas enzyme reads the genome. And if these enzymes can read more of the genome, it stands to reason that they should exhibit an increase in off-targets. So when we run this GuideSeq assay now using the same guide RNA as we did in that initial experiment with wild-type Cas9, what we see is the appearance of a few new off-targets with SPG and a few more off-targets with this SpRY enzyme. And if you look carefully actually at the PAMs shown here, hopefully you can see my mouse, of these expanded PAM enzymes, you can see that actually all of these new off-targets bear PAMs that are now accessible to these enzymes that weren't accessible before to wild-type. So essentially this shows that the expanded targeting potential of these enzymes does mean the potential for an increased off-target rate. But fortunately, a few years ago, we engineered these high-fidelity proteins of Cas9 that had improved specificity. This is very blurry on my slides. Hopefully it's higher resolution in what you're able to see. But this is published, so you can check out that study. Essentially, we engineered this protein called HF1 that had a few mutations in the Cas enzyme that improved its fidelity. And what we're able to do is take these HF1 mutations and plug them into both SPG and SpRY. And when we reran GuideSeq using these enzymes, you can see that now these off-targets that had appeared with SPG and SpRY now essentially are basically gone with the high-fidelity version of the protein. So two important things to note here, that at least by plasma delivery into cells, these high-fidelity variants preserve on-target editing, but importantly, they also eliminate off-target editing that results from this expanded targeting range. So hopefully these slides make it really clear that we have methods now to identify and monitor off-targets like GuideSeq, but now also methods to mitigate off-target editing like these high-fidelity variants that have become more important in these relaxed PAM variants that can spend more time searching more of the genome. Okay, all right, so I've got 10 minutes or 15 minutes and I'll tell you about some of the really interesting and useful applications that we've been exploring with these enzymes that have an expanded targeting range. All right, so essentially the question that we're asking is now, what can we do that we couldn't before? Why do we need these enzymes with expanded PAM compatibility? And I'll tell you, convince you hopefully of how these can be useful in various applications. And I think these expanded PAM enzymes are most immediately useful for applications like base editing, where you have to very, very carefully position your edit. So base editors, if you're not familiar with them, these are fusions to a Cas9 protein of a deaminase domain that essentially can catalyze nucleotide substitutions. But base editor or deaminase domains of the base editors can only function in a short sequence window that's dependent on a fixed distance from the PAM and how this deaminase is tethered to the Cas enzyme. So I direct you to the right side of this slide that essentially shows that at a certain distance from the PAM, the deaminase domain has an optimal activity. And as you move outside of that editing window, the activity of the deaminase decays relatively quickly. So the importance of the PAM at a specified distance from the edit that you want to make manifests very clearly in the base editor platform. So having enzymes that allow you to position the PAMs more carefully by being able to read more of the genome allows us to base edit previously inaccessible parts of the genome. So the question became, with these new enzymes, SPG and SpRY, can we make effective C to T base editors and A to G base editors, leveraging their expanded targeting capabilities? So here first I'm showing you data with the SPG enzyme compared to wild type. So each blue dot here is a different target site and base that is being edited. So you can see here we can more effectively edit many target sites that were inaccessible before with wild type Cas9. These are sites with NG and PAM, so we expect only about a quarter of those to be editable with the wild type Cas9 protein, which is why many of these are down around 0%. But now with the SPG protein, we see that the mean activity is actually quite high, and overall the editing efficiency is quite good with the SPG C to D base editors. We also tried SPG as an A to G base editor, where here again each dot represents editing of an A within a specific target site. And the vast majority of these high data points are A's that fall within the middle of the edit window, and some of these lower A's are ones that generally fall on the border of the edit window. But the take-home message here is that these SPG, CBEs, and ABEs are effective at making edits that we couldn't before with wild-type Cas9. We did an analogous experiment with SpRY, which is the enzyme that can recognize NR PAMs and NY PAMs. With CBEs and ABEs, again, both for the NR and NY PAMs, we see very effective base editing at sites that were uneditable before with wild-type Cas9. Okay, so to give you a real-world example of why this is useful, here is an example using wild-type SP-Cas9 as a base editor, where we tried to make a protective genetic variant where this edit could protect an individual against cardiovascular disease. Here, if we use wild-type SP-Cas9 with a C to T base editor, trying to change the T on the bottom strand shown in orange, what we found is that using the 3 guide RNAs that would position that C perfectly in the ideal edit window of the CBE, we see basically 0 editing of that target C base. And again, that's due to the fact that these PAMs are unreadable by the wild-type Cas9 protein. But when we use SpRY, which can actually read these NA PAMs very effectively, you can see with each of these 3 guide RNAs, we observe very good levels of target-based editing. And also, we do see some bystander editing of nearby cytosines. But fortunately, both of these bystander edits were either low level or in positions that make silent substitution. So using the SpRY protein, we could identify one condition where we achieve very high levels of intended edit and try to minimize any deleterious bystander edits as well. Again, this edit was impossible with wild-type Cas9. Okay, so hopefully that shows you and helps you understand how the SPG and SpRY-based editors can be useful to edit new sites in the genome. I'll now talk about some of the new base editing applications in the context of different diseases that we're working on with collaborators as well. So using some of these expanded PAM enzymes, this has accelerated a lot of our disease-focused research, where essentially we're working through this pipeline where we can use enzymes like prime editors to model these mutations in cells in a dish, and then we can also then go and correct them. If you're not familiar with prime editors, I encourage you to read some of these papers. They're actually quite exciting. Essentially, it's a fusion of a reverse transcriptase domain to Cas9 with an extended guide RNA that encodes a change in that extension. And the RT domain essentially can write that change from the extended guide RNA into the genome. So we can use these prime editors to effectively model many disease-causing sequences. So what we're able to do is build SPG and SpRY versions of prime editors and observed very reasonable levels of on-target prime editing with these enzymes, both at sites that are accessible with the wild-type enzyme, so having NGG PAMs, but also at sites that were essentially uneditable before with the wild-type PE. But now with both SPG and SpRY, we see very comparable levels of prime editing to what we see with wild-type on NGG, but now with SPG and SpRY on a site with an NGA PAM. And of course, like most prime editing, I think the efficiency on any given site can vary quite a bit depending on how you vary different parameters of the prime editor, like components of the PEG RNA and the PE3 nick and guide. Okay, so some of these expanded PAM variants paired with things like prime editors and base editors allow us to more rapidly iterate through this approach of modeling and correcting disease-causing mutations. We use prime editors predominantly now to make the disease models, and then we explore many different ways to think about remediating disease sequences like base editing, allele-specific knockout, and prime editing, all leveraging some of these new PAM variants. So one practical example here, where with a collaborator, we were able to make a cell line harboring this disease-causing arginine to histidine change. We did this using PAM variant prime editor. You can see here that we were able to make this heterozygous cell line that had one G allele and one A allele. But then using one of our PAM variant base editors, the ABE, we were able to essentially correct this back to wild type to near 100% efficiency in this experiment, showing that we can very quickly iterate through this pipeline now of making the cell line with the prime editor and using all these new relaxed PAM variant base editors we have at our disposal to explore therapeutic correction of this disease-causing sequence. With another collaborator, we were using a SpRY original 7.10 ABE to try and correct the disease-causing sequence. Here we selected two different guide RNAs that would position the edit window ideally over the target base. When we used the SpRY ABE with these two different guide RNAs, we observed relatively low editing efficiencies. But then when we used the ABE 8E version described by David Liu's lab, we observed a very dramatic increase in on-target base editing, in some cases more than tenfold, using the SpRY ABE 8E. So the improved catalytic activity of this engineered ABE domain allowed us now to make this therapeutic edit with greater than 50% efficiency in a largely unoptimized protocol. So I think this nicely shows how having some of these expanded PAM enzymes with these improved deaminase domains can enable very effective base editing applications. Okay, so I think all of that under the lens of increasing the addressable portion of the genome in terms of thinking about therapeutic genome editing. We're very, I think, keen on lots of different therapeutic strategies like base editing that allow us to make the exact corrective edit, allele-specific knockout for certain dominant negative diseases where we can differentiate wild type and mutant alleles, and then prime editing in cases where neither of these technologies are effective at making the change that we like. And we pursue both of these approaches, or all these approaches, with some of these relaxed PAM enzymes that I just described to you. But also in cases where we have enzymes with altered PAM requirements available, we use those because they're less likely to have off-target effects. And a couple really nice papers from David Liu's lab and from Beam Therapeutics lately showing, I think, the power of these PAM variants in base editing context for therapies. All three of these papers relied on PAM variants that allowed them to position the edit window of base editors as well. So if what I told you wasn't convincing enough, check out these papers because they also required PAM variants to make their edits of interest. All right, finally, one other application that I'll just briefly tell you about where I think a relaxed PAM enzyme is actually quite useful. And this is now no longer in cells, but in vitro. And I think hopefully everyone that's listening has used a restriction enzyme before. Essentially, these are enzymes that allow us to manipulate DNA in a test tube, modifying recombinant DNA nearly at will. One of the issues with restriction enzymes, though, is that they require a 6 or 8 nucleotide binding sequence, and that only usually occurs once or twice or 3 times in a plasmid. So we thought, now that we have this PAM-less SpRY enzyme, maybe we could change how we think about molecular biology, because we can cut anywhere in a plasmid. So some of the data that I'll show you essentially is an in vitro cleavage reaction with the Cas9 enzyme. Here I'm showing you data with wild-type Cas9. We take the purified protein, we throw it in an in vitro cleavage reaction on a substrate, and then we plot cleavage of that substrate or linearization of that substrate over time. So here with an NGG target, PAM target site, you can see that wild-type Cas9 very quickly linearizes that substrate, because we know that wild-type Cas9 can effectively target sites with NGG PAMs. And then these other 2 sites are largely inaccessible to wild-type Cas9. So it's just to orient you with this data. As we look across additional target sites, again, in vitro, now using guide RNAs targeted to the entire spectrum of possible sites in a plasmid, you can see again that wild-type Cas9 can very quickly cleave DNA with sites with NGG PAMs, but then is largely ineffective at editing these other sites, with the exception of some other PAMs like NGA or NAG, or a shifted NGG, which we know wild-type can target. But these other sites, or these other PAMs, essentially are uneditable by Cas9 in a test tube. But now when we use our SpRY protein, you can see that all of these target sites at the end of our digest are completely cleaved when we use SpRY. So the relaxed PAM compatibility of SpRY allows us to make an edit essentially anywhere in a plasmid in a test tube that we want. And as you think about molecular cloning and how right now you think about, the availability of restriction enzymes, we're sort of programmed into manipulating plasmids only in ways that we can do that when restriction enzyme sites are available. But now using SpRY, we don't need to think that way anymore, because we can cut a plasmid essentially anywhere. So to give you 2 concrete examples of why this is useful, one example here, we had an SA-Cas9 ABE8E construct in the lab, but we didn't have a P2A GFP version of this plasmid. And that's useful, obviously, if you want to sort transfected cells. So what we did is we essentially designed 2 different guide RNAs. You only need 1 because SpRY can make a double-stranded break. They cleave precisely at the end of this NLS. And what we're able to do is linearize that plasmid, use a PCR product to do a single-piece Gibson reaction or isothermal assembly reaction into that cut site to introduce this P2A GFP sequence. And we can do this with about 95% efficiency, which is consistent with the efficiency that you see with restriction enzymes. But now here we didn't need to rely on the availability of a restriction site right after this NLS, because we could cut precisely there with the SpRY enzyme. One other example I think that's really exciting in our own work, and taking this really full circle to our protein engineering, is how we can use SpRY protein to digest Cas9 anywhere that we want and build a library of Cas9 that's varied at any amino acid that we want. And how you do this is you take 2 guide RNAs, program them with SpRY, allow them to cut around the part of Cas9 that you're interested in mutating. So these amino acids are some of the ones that I showed you earlier that we varied historically to try and change the PAM requirement of Cas9. By linearizing the Cas9 plasmid at these positions, you can then order from IDT for a few dollars an oligo that encodes these degenerate nucleotides. So we can essentially do a saturation mutagenesis of parts of Cas9 enabled by this approach. So essentially, SpRY allows us to cut exactly where we need to. We can then use this oligo to clone in this library into Cas9. And you can see here that the signal, by Sanger sequencing this library of plasmids, completely deconvolves at these positions, which is what we want. This is showing that we've essentially built a library of Cas plasmids encoding all amino acids at these positions. So hopefully that highlights some of the utility of these enzymes. Again, to summarize what I showed you today, we've been able to engineer versions of Cas9 that can read more of the genome. And I showed you several different applications where they can be useful for making specific nucleotide changes by base editing, in the context of prime editors to make the models of disease-causing sequences, or even in vitro, being able now to cleave DNA anywhere that we want in a test tube. All right, for the sake of time, I'll skip to the acknowledgement slide to really acknowledge the people in the lab that are doing this work. This has been a huge team effort involving essentially everyone on this slide. So I'm obviously very grateful to everyone in my group, including our collaborators who've enabled a lot of these studies, and of course our funding agencies. I'll leave it on this slide. This was our recent lab retreat to Provincetown that we went on a couple weeks ago. Just to say that we're hiring, we're hoping to build the group at essentially every level, from tech to grad student to postdoc to lab manager. So if you're interested, or you know anyone that's interested in working in genome editing, technology development, and application, please reach out. So with that, I'd be happy to answer any questions that I can.
Okay, thanks, Dan, for the wonderful presentation. And we do have a couple of questions from the audience. So the first question is, like, if you have more usable PAM, and don't you also will increase the potential off-target cut sites? And I think you covered some of these information in the presentation, but if you want to explore, that'd be great.
Sure. Yeah, so I guess the question is, do we increase off-targeting rate? Yes. I showed that on some of the slides, that if we increase the off-targeting prevalence because these enzymes are reading more of the genome, I think that's essentially the expectation. So what I showed is we can mitigate these new off-targets by using these high-fidelity proteins that tune the enzyme to be more specific for its intended on-target site. I think also, you know, if you have a more specific version of Cas9, whether it's another ortholog that exists in nature, or an engineered version that allows you to access that PAM without needing these relaxed PAM variants, then obviously I would advocate in using the ones that search less of the genome because they're less likely to exhibit off-target effects.
Thank you very much. The second question is, would these SPG and SpRY enzymes also be amenable for epigenetic modification? And this would be potentially allow genome-wide specific silencing of certain genes.
Sure, I think that's a great question. So it's something we haven't formally tested, whether these SPG and SpRY enzymes can be catalytically inactivated and turned into activators or repressors or histone modifiers. I think it's a good question. I think, you know, it stands to reason that they should function in this context. We were able to make base editors and prime editors with these enzymes. I think you have to think carefully about the kinetics of these enzymes, though. So they're spending a lot of time searching a lot of the genome. So in some cases, we do need to express more of this enzyme if you want to achieve the same level of edit or epigenome edit. And just to say that more clearly, you know, SPG is searching 4 times more of the genome than wild type. SpRY is searching 16 or more times of the genome than wild type. So if you want to have the same on-target effects, depending on how you're thinking about your experiment, you might need to think more carefully about the amount of enzyme that you're able to deliver into the cell.
Thank you so much. And also the third question is, do you have any prediction for the accessible region of the human genome or other genomes, you know, in comparison with the SPG, SpRY versus the traditional SP-Cas9?
Sorry, predictions in the off-target effects, is that the question?
The prediction of the accessible genome, like the genome region, like how much percent of the accessible genome would be these different versions of SP-Cas9?
Sure. Yeah, I mean, I guess it depends on the genome of interest, but you know, the rough back of the envelope math is that with wild type Cas9, 1/8 of the genome is accessible if you assume an even GC content. With SPG, it's 4 times better than that. And this is, again, assuming both DNA strands. So for applications like base editors, the math is a little different because you need to target a specific strand. But with SpRY, you know, the vast majority of the genome is accessible within 1 or 2 bases. And I think, again, that's excluding certain things like repetitive regions of the genome or homopolymers that make it inaccessible. But yeah, some of these enzymes, especially in vitro and in, you know, certain types of human cell experiments, allow us to make edits to virtually any sequence that we want.
Got it. Thank you so much. And the next question will be, what conditions do you need for the Cas protein to function in vitro for molecular biology experiments?
Sure. So, yeah, the conditions are pretty standard, actually. I think our in vitro reaction conditions for some of the data that I showed you there's two different types of experiments. We've either expressed SpRY in a lysate from human cells, and that essentially the reaction's done in a lysate buffer and then a Cas9 reaction buffer. But with purified protein, I think the conditions, I want to say it's like a standard NEB buffer that they recommend for their Cas9 reactions too. So I think it's nothing really different than you would use for wild type Cas9 reaction buffer for in vitro cleavage reactions. It's essentially the same.
Got it. Thank you. So next one will be, in which cells has the PAM recognition been tested? And do you also plan to engineer your Cas to make it more accessible to closed chromatin regions?
Yeah, so we've tested these enzymes in my lab in a couple different human cell types. I think I've seen them used in plants, yeast, bacteria, all kinds of different organisms in other published papers. Some of our collaborators in a bioarchive paper, they used SPG and SpRY in C. elegans and in zebrafish. So I think these minimal PAM enzymes are extensible to other organisms. I think you have to think carefully again about the concentrations of these enzymes and how you use them, the temperature maybe of the other organisms that you're working in. But in theory, these are extensible to other systems.
Great. Okay, so we also
part of that was trying to engineer your Cas to make it more accessible to closed chromatin regions. Yeah, I think to the extent that the innovations have happened with wild type Cas9, I think it's an important and open question of how you can make these enzymes access other parts of the genome in non-dividing cells.
Yes, thank you. So the next one will be the enzymes for SpRY, and for instance, are they going to be commercially available soon? If so, are they going to be nearly as affordable as the restriction enzymes?
Sure. Yeah, I think, having started my lab 2.5 years ago, I can tell you we spent thousands of dollars on restriction enzymes. What we did is probably what most people do when they start their lab. If you're doing any molecular biology, you buy, I don't know, like your 40 or 50 favorite restriction enzymes and you fill your enzyme coolers with them. So the economics of whether these will be available commercially and what they will cost, I cannot tell you, but I can tell you the convenience of having one tube of an enzyme, and all you'll need to do if you want to target a different site is in vitro transcribe a new guide RNA. To me, that's very appealing. So the cost of in vitro transcribing a guide RNA, I should know this, but I mean, the oligo is like $4 or $5 from IDT. I want to say the in vitro transcription reaction is maybe $6 or $10 per guide RNA. So you're talking, I don't know, let's say $15 end to end per new site that you would target with SpRY. So I can't tell you the upfront cost of what the SpRY enzyme will cost to, buy it from a commercial vendor if they become available, but to access a new site, it's a pretty low cost. So hopefully that answers the question.
Thank you so much. And then one more question is, do we need like a specific gRNA design tool for these SPG and SpRY other than the traditional Cas9 design tools?
Yeah, that's a great question. So at least in the 175 sites that we've looked at in human cells, they don't follow the traditional guide RNA prediction algorithms very well. But I think that makes sense because those prediction algorithms rely also on the PAM sequence and the local sequence around the PAM. And the fact that these enzymes now are reading sites that were inaccessible before to wild type Cas9, which would score as bad sites, I think it makes those algorithms less reliable. So I think having new tools built on these scaffolds or other scaffolds of relaxed PAM variants probably are necessary for accurate prediction of activity in different organisms.
Thank you so much. Okay, so that's all the questions that we have today. And if you have any other questions in the audience, please feel free to email us and we will send over your and I really appreciate you attending this talk. And thank you, Ben, for the wonderful talk.
All right, thank you so much.